Hello, let us continue the survey of fungi module. Today's agenda will be to see agaricus or mushroom, which belongs to Basidiomycota. Before that, let us go through the usual exercise of seeing the root map of fungi module. First of all, Zygomycota. Zygomycota we have already completed through the life cycles of Rhizopus and Mucor. Then we started with Ascomycota. We have seen Saccharomyces, Aspergillus, Penicillium, Neurospora, Claviceps and even Pezaiza. After that, we have started with Basidiomycota. We have already completed Puccinia. Today's agenda will be to see Agaricus. After that, we will be seeing Stilago. Then we will be moving to fungi-like organisms, among which we will be seeing the phylum Oomycota. Within that, we have to see the life cycle of Phytophthora and Albugo. Then we will be seeing lichens, mycorrhiza, fungal toxins like general topics. So this is the agaricus or mushroom, which is a very common and familiar fungi to all of us because we normally eat mushroom, right? Then this is the structure. It is having a vegetative part called mycelia. Then it is having a fruiting body called what? This is the edible part. This is the mushroom that we eat. Okay, this is the basidiocarp. Okay, basidiocarp or the fruiting body which we eat. It is having three parts. It's having a stipe. Then it is having a pileus. Okay, pileus is this umbrella-like part. Then below which we will be having like gills. Okay, these curves called gills. This lines called gills. This is, if I am taking a cross section here, this is the gills. Okay, this is the gills. On the sides of it, you will be seeing the basidiospores. We will be coming to those parts. This is the gills. Okay, these are millions of basidiospores will be there here. Okay, you will see this. So this is about the life cycle of as agaricus. This is very simple life cycle. You know that there are two types of strains of basidiospores. Just imagine that when from a like when a basidiospore is released, there will be two types of basidiospores. We are not calling it as male and female, but we are just calling it as plus and minus because there is no sex organs as such in Basidiomycota as per agaricus. In agaricus, there is no sex organs. You cannot see any sex organs, but it is just through the fusion of two mycelia. So since it is fusing, normally fusion happens between opposite strains, right? That's the order of nature. Okay, opposite sexes attract. So like that, for that purpose only we are calling it as plus strain and minus strain. So we are calling it as plus spore and minus spore, from which plus strain and minus strain of mycelium arises. So there will be fusion. You can see that this can be fusion of two different mycelia. And before that, understand one thing. Like in some species of agaricus, there can be homothallic condition. That means both hypha will be of the same strain. Okay, in some species there will be heterothallic condition. There will be strain 1 and strain 2. So they will be fusing. So that just keep that in mind. That's just an additional information. Now they will be fusing each other, and you know that it was initially a monokaryotic type from one particular basidiospore, one particular type of nuclei bearing mycelium arose. Then this is from another basidiospore, another strain of nuclei bearing type of mycelium arose. When they fused, it became dikaryotic, right? Through mitosis division. Initially one cell was dikaryotic, then through mitotic division all the cells became dikaryotic. Okay, now by the aggregation, when this is just one type of fusion. Okay, this is one fusion only. That means one mycelium, second mycelium. So just one fusion we have shown here. Similarly, millions of hyphae will be fusing. Okay, and finally through the fusion, many millions of this particular hyphae will be fusing, and they will be forming a pinhead type structure. Okay, pinhead type structure below the soil. Actually, all these hyphae will be present below the soil, right? Just imagine that. Okay, we are when we are seeing some fungi or agaricus mushroom, we are seeing only this top fruiting body part. Remaining, like we are seeing normal grasses, because the remaining entire mycelium mat will be present within the soil. Okay, below the soil. So this is the aggregation of hyphae. All this joined together to form a pinhead like structure. Then it slowly started getting eros. Then it is slowly getting differentiated into a stipe, and then it's an umbrella like body. Then within that we will be having gills. Those gills will be bearing this particular spores. So let us see how that occurs. Okay, After this dikaryotic mycelium is formed, this pinhead like structure formed, and it's slowly differentiated into a thallus like this. This is a floating body. So this is a floating body. You can in exam hall you can draw like this. Okay, just show one basidiospore, two basidiospore, they got like mycelium got released, they got fused, and dikaryotic stage arose. Then you just show this type of a structure, which is very easy to draw. Then show a circle like this, like we are zooming into this particular area. Then show like this, just like this. Okay, it is also even the gills are also mycelium only, right? From you just show the aggregation of mini mycelium, then show that some bulbous part is protruding out. This junction we call it as sterigmata. Okay, we have seen already this thing in Puxine as well. So this, from this sterigmata, a bulbous structure will be released, and within that there will be fusion of the dikaryotic nuclei will be happening. We know that till here, till even in the gills you can see dikaryotic mycelia. But when the basidium is formed, this after this sterigmata there will be this bulbous structure is called basidium. Within the basidium like fusion happens. Okay, fusion happens. That means karyogamy happened. Then in this particular basidium you can see meiosis. So one particular nuclei, when it undergone meiosis, it released actually four haploid nuclei. Now there will be the formation of sterigma. Actually this is not sterigma, this is sterigma. Okay, from the basidium, at the tip of basidium there will be formation of sterigma. I am sorry, no, it's not here. Here, it's not here. From the gills there will be directly the basidium will be formed, and from the basidium there will be the formation of sterigma. This is the sterigma, and all the four haploid nuclei will be those created, that will be entering into these particular projections called sterigma. Okay, and at the end of the sterigma you can see what the haploid nuclei. That haploid nuclei will be accumulating some cytoplasm around itself, and a wall will be formed around itself. So that will be converted into a what? Basidiospore. Now this basidiospore will be released and cycle continues. Easy to draw, right? Very easy to draw. So this is another kind of representation. Same thing only. Two type of mycelium got fused, and within the basidium there will be fusion of zygote. Zygote undergone meiosis, four haploid nuclei. Then there will be forming basidiospores that will be released. Then plus and minus strain type of mycelium will be formed. They will be fusing again. This particular type of this, what is this thing? Dikaryotic hyphae, right? This dikaryotic hyphae will be lining the gills. Okay, if I am taking one particular hyphae, you can see that many such hyphae will be aggregating together and it will be forming a pin-like structure. This pin-like structure will be elongating and differentiating into a basidiocarp. So this is another type of representation. All things show the same thing only. Okay, same thing only. Just for your clarity, I have attached these things. Okay, you know that. Yeah, what is it? Yeah, one basidiospore and another type of basidiospore. They formed two types of strains of plus strains and minus strains of mycelium. When many such mycelium fuse, that they aggregated together, a button type structure is formed or a pinhead type structure is formed. It will be slowly coming around like on the top of the ground. That will be differentiating into a stipe, and it will be forming a pilus or head. Then it will be having gills within at the bottom of the pilus. Then that will be having projections. See, this is the this particular gills area. If I am zooming in, you can see that it will be protruding out basidium. This is the basidium which is protruding out from this particular gills area. This is the initially dikaryotic stage. Then fusion occurs. So zygote is formed. Then meiosis. Four haploid nuclei will be released. Then there will be projections called sterigmata at the tip of which this particular haploid nuclei will be coming. Then it will be forming itself into a basidiospore. Then slowly it will be released. Okay, and so this is the structure. This protruding structure is called basidium. From the gills you will be seeing basidium. At the tip of the basidium there will be sterigmata formation. So at the tip of the sterigmata the haploid nuclei will be coming and it will be converting into a basidiospore. And when will this structure will be formed? This fruiting body will be formed during the winter season. Okay, you know, during winter season there won't be sufficient temperature or nutrition available. So to overcome that, this structure, this particular like basidiospore has to be released. Okay, because the basidiospore will be laying as such without germination till the winter season is over, this mycelium has to get the nutrition, right? Only in the spring and summer season you can get the nutrition. So to overcome the winter season, this particular mycelia will be forming a structure called the fruiting body called the basidiocarp, which will be releasing the basidiospores, which will be helping it into like overcome the winter. So this is the life cycle. You can just see that there were two types of spores, then two types of mycelium, then somatogamy, that means fusion of this mycelium, then plasmogamy, fusion of the cells or of this particular mycelium. Then it formed a dikaryotic stage, then dikaryotic cell that undergot mitosis. So every cell in the mycelium became dikaryotic, so that will be forming the secondary mycelium. Then what is rhizomorph? Rhizomorph is the pinhead like structure that is formed. Then that will be differentiating into basidiocarp. Then basidiocarp, within the basidiocarp you can see gills. Within the gills at the protruding end will be called basidia. From basidia you can within which basidia meiosis happens. Then again basidiospores will be created. This is the same kind of representation again. Plus and minus type of spores, like dikaryotic formation, then pinhead like structure, then differentiation. So this is all. So I have attached multiple type of figures. You can so that you can get a utmost clarity, or these are easy to draw diagrams, right? So just for you to easily draw, just show that this type of a pileus or the basidiocarp you just show, then zoom in with an arrow, then show that at the tip of the mycelium of the gills there will be basidia formation. So it is also in the dikaryotic stage. Now it will be fusing. Fusion occurred or karyogamy happened. Then sterigmata formation. Okay, in the tip of the basidium. At the tip of sterigmata there will be ascospores. So this is the knot. You can take down the knot by forming by posting the video. You know that, okay, as Agaricus, some of the species are edible, some of the species are actually poisonous. Okay. These are the two common species that you will be studying. Okay, Agaricus campestris, which is known as field mushroom, Agaricus bisporus, which is the normal white mushroom that we see. Those are edible. Then one poisonous variety, if you want to know, that is Agaricus xanthoderma. Okay, just know these names. So these are the same things. The structure, if you want to know the structure, take down the knot, pause the video. Then this is the vegetative structure. I have already told you what is this thing. So what is primary mycelium? The first mycelium that is developed from the spore, that is primary mycelium. Then the secondary mycelium will be the dikaryotic mycelium. The tertiary mycelium will be the mycelium that is present within the gills and all, right? It will be differentiating into a fruiting body. Within that there will be bikaryotic mycelium. From the bikaryotic mycelium from the dikaryotic mycelium within the gills there will be formation of basidium, right? So that mycelium which is formed towards the end, that dikaryotic mycelium is known as tertiary mycelium. That will be giving rise to fruiting body or basidiocarp. So about the reproduction, I am just attaching the knots here. Okay, vegetative. There is three ways. Okay, vegetative through fragmentation. If one particular portion of the mycelia is got fragmented, that will be giving rise to new thalli. Then asexual through chlamydospores, and sexual that we have already seen, plasmogamy, karyogamy, etc. Okay, through somatogamy. So chlamydospores is not so important for us, so that's why we will be concentrating on sexual reproduction. So this is the thing. I have explained everything. Pause the video, take down the knot. First, the plasmogamy or the somatic fusion will be happening between two opposite strains. Then this is the thing. Actually, you need not worry about this drawing. It will not be asked. Okay, just for your understanding, a button type or pinhead type structure, it will be slowly differentiating into a basidiocarp. Just show this thing. Then zoom in. So at the tip of the gills, you can see basidium. So what will happen? Karyogamy will be happening. Then that will be leading to the formation of sterigmata and then basidiospores. This is the basidium within which karyogamy happens, then basidiospores. So karyogamy, then meiosis. So we wind up Mycota genus called Agaricus here. So I hope you have understood and you have posted the video and taken down the notes. So we are winding up here. Next we will be seeing Ustilago. With that we will be winding up Basidiomycota. Then only thing remaining will be the fungi-like organism Mycota, two genus within that. Okay, Phytophthora and Albugo. With that we will be finishing the entire life cycles of fungi. Thank you.